लास्ट क्लास में हम SNP तक डिस्कस किया था. नाउ विल मूव ऑन टू फंक्शनल जीनोमिक्स. See, this tells, or this part of the genomics tells us about how the gene produces their products and how these products are actually interrelated and how do they function. 2 or 3 products, how do they interact. Okay, all these things are being discussed in functional genomics. The first topic in this in your syllabus is fish. Fluorescent in situ hybridization. In situ means in their natural site, मतलब वो जहाँ पे वहाँ. So this is one type of hybridization. As you all know, hybridization में हम एक original DNA में probe को hybridize कर रहे हैं. Probe is labeled, so we can easily identify the probe, so we can identify that gene. Now if you do this within the cell, inside the nucleus, okay, then this is known as in situ. Normally हम इन DNA को बाहर लेके आते हैं एक जेल में separate करके, then they are fluorescently labeled, or then they are identified using fluorescently labeled probes. But in actually fish, we send the probes into the cells and we identify the actually that particular gene inside the organism or in their own original place. This is the word in situ. The rest of the thing is simple hybridization only. So let us see what are the major requirements for this fish. First is probe. As you all know, probe is a single stranded DNA which is labeled. The question is how to label DNA, that too fluorescently. So for this fluorescent labeling, Rigby and Paulberg, they have come with a very interesting technique, and that is known as nick translation. By nick translation we actually label probes. Okay, so this is a prerequisite of fluorescent in situ hybridization, because we need to have probes with us. So probes are actually labeled by using nick translation. Let us see how this nick translation is done. This is done with the help of 2 enzymes. Suppose this is the DNA which is unlabeled, okay, which is not labeled. Now first we treat this with an enzyme which is known as DNA. DNA is 1. DNA is 1 will make some single strand cuts on this DNA. They make some single strand cuts. These single strand cuts are known as nick. Now what happened, the another enzyme which is DNA, the next enzyme involved in this is DNA polymerase 1. आपको पता है इसका काम है म्यूटेशन रिपेयर में. So what actually this DNA polymerase 1 does is they remove this, actually they remove this particular part or they seal this particular part, or they remove this particular part and add DNTPs to that. Let us see what happens here. सपोज हम इस स्ट्रैंड को देखते, इस स्ट्रैंड को निकाल के अब नया स्ट्रैंड यहाँ बनाएंगे DNA पॉलीमरेज. जब ये नया स्ट्रैंड बनाते हैं, देन वी विल गिव इन द मीडियम fluorescently labeled DNTPs. DNTPs मतलब AGTC, which are given in the medium, they are labeled. So once this part is again they are made again, they will incorporate these DNTPs which are labeled. That makes probes labeled. It can make ये स्टैंड भी नया बना सकते हैं और इधर भी नया बना सकते हैं. What happens? That particular part gets labeled. This is how the DNA gets labeled. अब हम देखते हैं कि फिश, अब इस लेबल्ड DNA से और प्रोब से हम एक जीन को यदि हाइब्रिडाइज करके देख रहे हैं, that is fluorescent in situ hybridization. We will take one example and study the application of fish or the use of fish. There is a disease known as CML, chronic myelogenic leukemia. Leukemia is cancer. This is chronic myelogenic leukemia. Okay, leukemia is a type of cancer, and this is actually due to a chromosomal mutation, that is translocation. Translocation means two chromosomes which are non-identical, they exchange parts between them. This is known as translocation. Let us see what is the reason behind the disease CML. There is a translocation between 9 and 22nd chromosome in human being, leads to the disease chronic myelogenic leukemia. You know, 9th chromosome is a bigger one, 22nd chromosome is a smaller one. If they exchange parts between them, okay, there may be again some more difference in their size. This results into more longer, larger 9th chromosome and more shorter 22nd chromosome. This shorter chromosome is known as Philadelphia chromosome. Philadelphia is a place, and Philadelphia chromosome is an indication of translocation between 9 and 22nd. But how come we confirm? Actually इसमें confirmation कैसे लेंगे? ये Philadelphia chromosome बने हुए हैं between the translocation between the 9th and 22nd chromosome. इसको confirmation हम करते हैं by using fish. तो हमें दो जीन्स का नाम याद रखना है. पहला जीन है हमारा BCR जीन, सॉरी ABL जीन. पहला जीन है हमारा ABL जीन, which is on 9th chromosome. नेक्स्ट जीन है हमारा ABL हो गया, उसके बाद BCR जीन. नेक्स्ट जीन है BCR. अगर आपको नॉर्मल केस में if you use proof to label CABL, that is ABL gene, you will get 9 chromosome in red color. And if you label the probe of BCR, you will get on 22nd chromosome a green color. This is normal case. But what happens after translocation? देखते हैं translocation कैसे हो रहा है. Translocation actually happens BCR जीन के नीचे से कट करके ये पोर्शन इधर आते हैं, इसके पोर्शन इधर से कट के उधर चला जाता है. This will result in such a way. I'll show you. इसमें रेड कलर का जीन और इसमें ग्रीन कलर का जीन। सो व्हाट हैपेंड, इस पोर्शन जब निकल के इधर चला गया तो इसका लेंथ बढ़ गया, इस पोर्शन यहाँ से वहाँ चला गया तो इसका लेंथ शॉर्ट हो गया। बट व्हाट हैपेंड, ABL एंड BCR जीन दोनों 22nd क्रोमोसोम में आया। अब हम यहाँ देखेंगे तो इस क्रोमोसोम में कोई कलर नहीं होगा और इस क्रोमोसोम रेड एंड ग्रीन पास में आता है तो हमें येलो या ऑरेंज कलर में मिलेंगे। सो इफ़ यू गेट एन येलो या ऑरेंज कलर ऑन द क्रोमोसोम नंबर 22, this indicates that there is a translocation between 9 and, अगर 9 and 20, क्योंकि हमने ABL जीन एंड BCR को लेबल किया, ABL 9 क्रोमोसोम का है तो 9 क्रोमोसोम के ABL अगर 22nd में आ गया इसका मतलब इन दोनों के बीच में हुआ है। और जब हमने बहुत सारे सेल देखा उसमें ज़्यादातर सेल में हमें येलो कलर मिला, ओके, देन वी कैन से दैट द सीवियरिटी ऑफ डिजीज़ इज़ मोर। अगर एक ही दो सेल में मिला तो अर्ली स्टेज में है, कुछ ही सेल्स में ट्रांसलोकेशन हुआ है। सो दिस इज़ हाउ fish is being done, application of fish in fluorescent in situ hybridization in confirming the disease chronic myelogenic leukemia.